Hello, everyone. Thank you for joining us. I'm Danielle Goldberg, Senior Manager of Product Management at Illumina, and I'm thrilled to be joined by Fiona Caper, our VP and Head of Assay R&D. We're excited to introduce you to one of Illumina's latest innovations, the 5-base solution. Over the next 30 minutes, we'll delve into the key technological aspects and discuss how our innovations are set to unlock dual omic insights more quickly and easily than ever before. Before we dive deeper into the 5-base solution, I'd like to clarify what we mean by 5-base. DNA is inherently multiomic, with molecular information stored as both genetic and epigenetic bases. The genome is made up of 4 canonical bases, A, T, C, and G. However, methylation adds an additional functional layer to cytosine, effectively creating a 5th base. Methylation is often referred to as the 5th base of DNA because 5-methylcytosine is a chemically modified form of cytosine. This modification plays a crucial role in gene regulation without altering the DNA sequence itself. Cytosine methylation is dynamic and can change over time due to environmental exposures, lifestyle factors, and disease. Researchers have discovered that abnormal DNA methylation is linked to many diseases, including cancer and neurodevelopmental disorders, making it an emerging class of biomarkers. Now let's introduce you to our 5-base solution. At Illumina, innovation is in our DNA. Our R&D team has spent years developing innovations that collectively unlock the most accessible 5-base genome. Our enzyme engineering team has created a novel enzyme that directly converts methylated cytosine to thymine, preserving genomic complexity and information. Our bioinformatics team has developed novel algorithms powered by Dragon and our new Illumina connected multiomics infrastructure, enabling ultra-fast and simple analysis. Our systems engineering team has integrated these innovations into a seamless workflow, leveraging our leading systems to deliver unmatched scale and precision. Together, these innovations will unlock 5-base insights that are simpler, more scalable, and more affordable than ever before, making dual insights within reach. Current industry constraints in sequencing the 5th base include cost, limited scale, complex workflows, and analysis overhead. Our 5-base solution aims to eliminate these constraints, making dual insights more accessible. It will provide the affordability, simplicity, and scale needed to maximize dual omic discovery. Library preparation will take less than a day and support various sample types, including cell-free DNA. Illumina will provide both whole genome and targeted enrichment solutions to ensure study flexibility. The workflow extends from library prep through analysis, offering an exceptionally simple and streamlined process that reduces complexity and overhead. With the Illumina 5-base solution, we anticipate the ability to run up to 96 5-base genomes per NovaSeq X run. Dual analysis is made easy with Dragon, providing a single output with accurate genetic variant and methylation calls, and Illumina connected multiomics, providing a simple user interface and multiomic data visualizations. Overall, the Illumina 5-base solution will deliver the affordability, flexibility, and simplicity needed to unlock deeper biological insights at scale. The Illumina 5-base solution offers a streamlined workflow from library prep through analysis for simultaneous methylation profiling and variant detection. Starting with library preparation, Illumina's proprietary single-step enzymatic conversion and optimized library prep chemistry and protocol provide a simple initial step in the NGS workflow. Library complexity is preserved, resulting in greater mapping efficiency and maximizing sequencing data yield. Integrated Dragon analysis performs alignment and variant calling, as well as methylation calling, delivering high accuracy dual outputs. I'll now hand it over to Fiona, who will walk us through the underlying innovations in more detail.
Thanks, Danielle. Illumina 5-base technology is based on an enzymatic conversion process that specifically converts methylated Cs to Ts. So today's gold standard technologies like bisulfite conversion and the enzymatic EM-seq method, they convert unmethylated Cs to Ts. And because the vast majority of Cs, around 95% of Cs, are unmethylated, these technologies end up converting almost all Cs in the genome to Ts. And as a result, you end up with a library that has what we call 3-base complexity. So that means that most of the bases in the library are A's and T's, and then very few G's and C's. And these library types are harder to sequence, so they're more difficult to map. And in the case of bisulfite conversion, the DNA is damaged in the process. If you wanted to assess both genetic variants and methylation of the same sample, you would have to make 2 separate libraries, one for the assessment of methylation status, and then one for genetic variant detection. Now, Illumina 5-base technology, on the other hand, converts only the methylated Cs to Ts. And so you therefore maintain that 4-base library complexity because only a small subset of the Cs are now converted to Ts. And as a result, the libraries are easier to sequence, they're easier to map back, and very importantly, one can now generate this one single library and detect both methylation and genetic variants. Illumina 5-base technology is based on a single-step enzymatic conversion that is added to our standard library prep process. The enzyme is a bespoke enzyme that was entirely engineered at Illumina, and the protein engineering team screened millions of mutants over multiple rounds and selected mutations based on high selectivity for methylated Cs, high efficiency of conversion, and high stability of the protein. And additionally, because the assay delivers a new data type, this 5-base data type, we also generated new algorithms, and these have been integrated throughout our software ecosystem for both secondary and tertiary analysis to fully realize the potential of 5-base technology. So substantial updates across the Dragon pipelines boost data yield and provide accurate genome and methylome annotations for germline and somatic applications. We deliver higher coverage and a more complete genome with multiomic annotations 4 to 10 times faster than is possible with traditional methylation sequencing methods and alternate pipelines. But the discovery doesn't stop there. The Dragon genomic annotations can transfer seamlessly into Imagine and ICI to interpret and prioritize variant calls. And we are integrating methylation analysis tools into Illumina connected multiomics to identify and characterize biologically driven changes. Illumina 5-base technology delivers both high accuracy methylation detection that is on par with today's gold standard bisulfite conversion and the EM-seq methods. That's demonstrated here on the left with methylated POC19 and unmethylated lambda. And genetic variant detection accuracy that is vastly superior to existing methylation detection technologies and very close to the performance of standard whole genome sequencing methods demonstrated in the middle panel. The ability to detect methylation and genetic variants together in a single assay delivers a 5-base genome, providing dual omic insights.
Thank you, Fiona, for sharing more details about our cutting-edge advancements. It's thrilling to see how years of innovation have led to exceptional data quality and dual omic insights. We've also had the privilege of involving some of our customers in this journey of innovation. At the recent Advances in Genome Biology and Technology conference, Dr. Beckham Sidikovic from London Health Sciences Center presented a case study on detecting genetic and epigenetic signatures in individuals with rare diseases. Patterns of DNA methylation can act as biomarkers to help classify variants of unknown significance. Sadikovic's lab developed EpiSign software that can accurately identify a growing number of episignature disorders. His presentation showcased the power of the 5-base solution, a single assay capable of accurately detecting dual signatures of disease. Looking ahead, he mentioned that rare genetic disorders are just the beginning of what can be uncovered with dual omics. Overlying the genomic and epigenome reveals biological insights into health and disease, encompassing environmental exposures, lifestyle factors, and conditions such as cancer. We are excited about the potential discoveries that can be made to improve human health using the Illumina 5-base solution. Multiomic approaches that combine methylation profiling with genetic information can connect functional layers, illuminating complex pathways of development and disease. The Illumina 5-base solution, which provides accurate genetic variant and methylation detection in a single assay, has the potential to significantly advance disease research, therapeutic development, and ultimately improve human health. We are eager to offer this solution to the scientific community in the near future. Thank you very much for your time today. We hope you share our excitement about the upcoming 5-base solution. And we would like to now invite some of our inventors for a brief Q&A session. I'm joined here today by Sergio Pizakovic, our VP of Head of Protein Engineering and Molecular Sciences and R&D, Eric Brewstead, our Associate Director of Scientific Research and Molecular Sciences Department, Suzanne Rohrbach, Senior Staff Bioinformatician Scientist, and Fiona Caper, our Head of Assay R&D. Thank you all for joining us here today. There's been a lot of buzz around this new 5-base solution. So I'd like to start off by asking, why are you so excited about this technology and why do you think it's important?
So maybe to begin with, the reason I'm the most excited about this is that for the first time, we are offering customers the ability to do something that until now was impossible. In essence, this technology allows our customers to look at epigenomic information at the same time that they look at genomic variation. And this is a capability that was simply not existing in the market. Until now, customers will have to do one thing or the other and then combine the data if they wanted. So bringing those two things together, we think is super exciting. And then besides, the richer biological information they get, the workflow is super simple. It's less than a day from sample to sequencing. And we think it's going to also be a great price point. It's going to be cheaper because instead of having to do this twice in a single run, customers will be able to take the full benefit of our technology.
I'm probably going to reiterate some of the things that Sergio just said. So I'm very proud of what the team has accomplished and very excited to represent what we've built together. It's not an iteration of a protocol that's available today. It's really testing new ideas. How do we improve methyl-seq assays? How do we reimagine what the bounds of a genome are? And how can we provide an inherently multi-omic readout of DNA? Because DNA is multi-omic. The methylome and the genome, they're physically linked to each other, both through the covalent bonds, but also because the methylation influences the outcomes of each other's variations. So this technology gives you a readout of both, but not as a standalone assay, as Sergio already said. And then we're delivering an end-to-end solution. So it goes all the way from sample preparation through sequencing, and then at the back end, the data analysis that drives these deeper discovery across many different disciplines.
Thank you so much, Sergio and Fiona. Can you explain the conception and development process of the 5-base solution?
Absolutely. So I'm going to start with a foundational idea. This is an idea, actually it was Eric's idea, so it's awesome that he's here with us today. It's a foundational idea that basically the concept is that he thought of a way to engineer an existing enzyme to enable that enzyme to do something that doesn't occur in nature. I'm not going to go into the details of that, but basically by engineering this enzyme in this very tailored way, we can now basically enable our customers to look at both methylation and genomic variation at the same time. Now, of course, an enzyme is not enough to have a product, so you need, and this is the beauty of Illumina, where you bring expertise from all across the board, from protein engineers like Eric, all the way to assay development and informatics. And I also think it's great that we have Suzanne here because she led that informatics effort as well. And the combination of all those three pieces is really what enabled our technology to work the way it does.
That's very exciting. So maybe I can turn it to Eric. Eric, can you give us a bit more context around how this technology improves upon existing methods?
Yeah, I'd be delighted to. The key innovation to our technology is a bespoke engineered enzyme that we developed in-house at Illumina by our protein engineering and assay research and development teams. We designed this enzyme to be selective for methylcytosine, and this allows us to directly target and tag methylcytosines across the genome. And it's this unique enzymatic selectivity that really differentiates us from most on-market products, and those typically target normal cytosine. Right. So as a result, this technology will enable our customers to accurately map methylation across the genome at single base resolution. And because we don't target unmethylated cytosine, we can also get genomic variants within that same single simple workflow. Right. So this will provide our customers with much more comprehensive and detailed insights into genomic information by providing both that epigenetic and genetic patterns and variations that you typically would use multiple assays to achieve.
I can tell you're all quite excited to bring these innovations to customers, as am I. So maybe I'll turn it to Fiona. Fiona, can you describe how all of these innovations come together to create a complete assay and workflow for our actual users?
Sure. Yeah, it's actually very simple. So this specific technology looks a lot like a typical whole genome sequencing library prep. So you start with your DNA, then you do your standard end repair, A-tailing, and you ligate on your sequencing adapters. And then after that, you denature the DNA, and then you perform this additional 30-minute incubation with our new enzyme. And after you've done that, it goes straight into the indexing PCR. And at that point, you're ready for the cleanup, quantification, and then you put it on your flow cell. So it's really very simple. It's just less than 30 minutes of additional hands-on time. It's just that one incubation. There are no toxic chemicals, so you can just do it on the lab bench. There's no excess DNA damage. And so basically the entire process from DNA to putting it on the sequencer is less than 7 hours. So it gives you a lot of flexibility in your workday, and it's very scalable for large cohorts. And then really, most importantly, can't emphasize this enough, you only need to do one library prep per sample to interrogate both the genetic variants and to detect methylation. Now, additionally, this is a very modular technology. So that means it is also compatible with enrichment. So there's no need to buy expensive methylated adapters. And it's completely compatible with our very efficient hybridization capture method, which adds less than 5 hours of prep time. And then the assay is compatible both with genomic DNA that has been shared, but also with low input cell-free DNA. So now it can support a very growing diversity of research objectives.
That simplicity and versatility are powerful benefits. We've been focused mostly on the upfront sample preparation, but Illumina technology obviously extends to further steps. So maybe Suzanne, you can address this question. Are there benefits of the Illumina 5-base technology for sequencing?
Yes. So only about 5% of cytosines in the genome are methylated. So that means that methods that convert the unmethylated positions are pretty much erasing all of your Cs and leaving you with just a 3-base genome of A, T, and G, which causes challenges for things like color balancing and clustering and generally lowers your data output. But because we're converting rare methylated cytosines, our libraries look pretty much the same as an average DNA sample, which means they play well with our sequencers. You don't need to add a bunch of phi-X, flow cell capture and clustering efficiency is high, and base call quality is reliable across many cycles. Second, our assay is gentle on the DNA, and that's a big benefit for low input samples that are sensitive to numerous cleanup steps and harsh chemical treatments. So with 5-base technology, you don't need to worry about whether you still have enough molecules left when starting your sequencing run. And both of those factors carry over into analysis. Having a complex library with minimal base conversions means higher alignment, more uniform and complete genome coverage, and better sensitivity for detecting differences.
So Suzanne, you just mentioned that this assay's benefits improve analysis. The analysis part of the workflow can be challenging to navigate for many researchers. What informatics solutions are we providing to better enable our customers throughout the entire discovery process?
Yes, software is integral to our 5-base technology, and I am so excited to share the features that we've been working on for the past 4 years. Our team has partnered with the Dragon team to make our 4-base genome analysis software methylation aware. And this was no small undertaking. We've touched nearly every step of that pipeline, creating 5-base alignment, which provides a 98% mapping rate and supports the multi-genome mapper, integrating automated QC reporting for confidence in every sample, extending UMI processing to produce single molecule methylation calls, and custom building out novel algorithms for sensitive germline and somatic small variant calling. And all of that processing runs in under an hour on a 30x genome. From there, we've been collaborating with the Illumina Connected Multiomics software group to build out capabilities for the data interpretations that power discovery. So we're introducing new tools to identify regions with differential methylation patterns between groups, and then easily plug those features into clustering and interpretation algorithms such as UMAP and gene set enrichment, or put them into regression and classifier building to understand the extensibility of novel findings. So all of these solutions are going to offer a lot of flexibility to deeply explore the 5-base genome, but with a straightforward interface that's making multiomics more accessible.
Fantastic.
Danielle, I actually have a question for you now. So what are the key benefits of the Illumina 5-base technology for our customers?
Thanks, Fiona, for asking that. So I'm very excited about this solution, and some of the benefits are to our customers is that Illumina has consistently made their innovations more affordable over time. And this new solution really combines insights that previously required 2 separate assays and 2 separate sequencing runs. So we can save time, costs, and effort and get dual insights from a single workflow. Additionally, it offers cost savings in the data yield, as alternative methylation sequencing methods often result in wasted reads due to poor alignment and DNA damage. And our new technology will eliminate these issues. So some of the key benefits are that we get deeper genetic and dual omic insights, a simpler workflow, and cost savings. And these advantages really make the technology accessible and valuable for a wide range of applications. Well, thank you everyone here for your valuable insights and contributions to today's discussion on the 5-base technologies. Your expertise and innovative perspectives have highlighted the potential and future applications of this groundbreaking technology.